Hello everyone, my name is Joel Casillas Gonzalez, and this is my Bio 282 lab final presentation. In this presentation, I'm going to compare the accuracy of the news article scientists are just beginning to understand mysterious DNA circles common in cancer cells compared to its source material. Circular ecDNA promotes accessible chromatin and high oncogene expression. In the news article, scientists are just beginning to understand mysterious DNA circles common in cancer cells. The main takeaways are that extrachromosomal DNA and are extra circles of DNA that float alongside chromosomes. As you can see in this illustration, the ecDNA are right next to the chromosomes. ecDNA is common in cancer cells and plays a major role in many cancers. DNA in chromosomes are packed tight into spools, meaning that all the DNA in these chromosomes are very, very compact. DNA from chromosomes can be copied into ecDNA, allowing it to be more accessible to the cell's machinery. Large inheritance of ecDNA can speed up cell growth. For example, cancer may become more aggressive, and healthy cells also can have ecDNA. All this information is from Zimmer, the author of this article. As for the primary literature, circular ecDNA promotes accessible chromatin and high oncogene expression. This is the summary. Oncogenes are encoded on ecDNA are amongst the most highly expressed in the transcriptome of tumors. Human cancer cell lines and samples from the Cancer Genome Atlas were investigated through whole genome sequencing, amplicon architect, and fluorescent in situ hybridization of tumor cells in metaphase. The circular structure of ecDNA was reconstructed. Amplicon architect is a computational tool that can classify DNA structure as either linear or circular. The fluorescent dye helps scientists determine ecDNA shape with a microscope. Circularization of oncogenes leads to increased numbers of RNA transcripts. When measuring the number of RNA transcripts, extrachromosomal DNA, which is circular, produce more RNA transcripts than linear chromosomal DNA of the same gene, even when normalized to the same gene numbers. Chromatin organization increases accessibility to regulatory machinery of transcriptase. Researchers of this paper use ATAC-C to visualize accessible chromatin directly. This showed the lack of long fragments in ecDNA, which indicates that ecDNA is more accessible than chromosomal DNA because its nucleosomal organization is less compacted. As we can see in this graph, ecDNA is in red. Most of its fragments are less than 500 base pairs as compared to chromosomal DNA, the gray line. Some of its fragments are more than 2,000 base pairs. Researchers also demonstrated that ultra-long ridge chromatin contacts could have some effect on distal gene expression. All this information is from the paper as cited below. The graph was recreated from the source material made by me. As for the accuracy of this news article overall was accurate. For example, healthy cells do have ecDNA as stated in the primary literature when they were talking about the differences in ecDNA and healthy somatic tissues. ecDNA is more active in chromosomal DNA. This was explained in the paper by ecDNA structure and less compaction of chromatin. But often the article lacks further explanation or was oversimplified. For example, the article starts off by stating that ecDNA is more circular without any further elaboration, how and why they are circular. The fact is just stated, while in contrast, the primary literature goes in depth with multiple approaches how the circular structure of ecDNA was determined. Also, the article oversimplifies why ecDNA is more accessible to the cell's machinery because they do not talk about how chromatin in ecDNA is not as compacted as linear chromosomal DNA. Explanation. The reason for the article's oversimplification of information from its source material is due to the nature of articles and who it caters to. It is in the nature of articles to be brief, to communicate as much information in as few words possible to a short attention span audience. The audience of articles are also not made up of scientists. Most of the audience does not have the foundational knowledge to know technical terminology and processes that are used in research papers. Using technological terminology and processes would only work to confuse the readers. This is why information is simplified to communicate effectively and broaden its audience. These are credits made by me in the context of Bio 282 and references.